For this topic, we'll be focusing on Streptococci and Enterococci isolation and identification. Now, just a brief overview regarding these two organisms. Your Streptococci and Enterococci, they differ from the Staphylococci in two significant characteristics. First, most isolates, they occur in chains. They are usually known for having long chains rather than in clusters. And they also lack the enzyme catalase. So, no catalase. Therefore, ang reaction nila in the catalase testing, they are negative. Catalase, remember, this is an enzyme that degrades hydrogen peroxide to form bubble formation, your oxygen, and water. As compared with other gram-positive cocci, the cells of the Enterococci and some Streptococci, they appear more elongated rather than spherical. Now on this table, it shows the different types of categories where they are classified into. They can be classified as to a Lancefield group. This classification scheme was developed back in the 1930s by Rebecca Lancefield. They are also classified whether if they have alpha, beta, or gamma hemolysis. And we also have here the common terms where they are known for and the different disease associations. Now, for us to better understand or to appreciate the hemolytic patterns further, we have the following images. For beta hemolysis, always remember that for beta, there are clear zones. No? Kaya yan clear because the organism was able to lyse the blood present in your agar. As for the alpha hemolysis, it's kind of greenish. No? There is a discoloration that happened on the areas where our bacteria grew. And as for the gamma hemolysis, you're just going to see there the growth of the colony, but there is no lysis, no hemolysis pattern, no change in your agar. Now, let's discuss them further. We have first here the beta hemolytic groups. Under this, we can categorize your groups A, B, and then C. There are also a few species under group D, which are beta hemolytic. However, they will be discussed under the alpha hemolytic section because they are variably hemolytic. Now, the main representative of your group A Streptococci, that is by far the most serious. It can rapidly injure cells and also tissues. What is that organism? We have your Streptococcus pyogenes. Again, Streptococcus pyogenes is under your group A Streptococci. When the colony of this organism grows on a blood agar plate, it will produce clear zones. Therefore, we term that as a beta hemolytic organism because the clear zones usually it is 2 to 4 times larger than the diameter of the colony. As for the morphology, these bacteria are still spherical in shape. So these are cocci still. They are arranged in short chains, in clinical specimens, and longer if they are grown in broth. Now we have your group B Streptococci, which is your S or Streptococcus agalactiae. This organism is an important cause of serious neonatal infection which involves sepsis and also meningitis. And actually in the adult population, S. agalactiae infections consist of abscesses, what else, endocarditis, even septicemia, bone, and soft tissue infections, as well as pneumonia. Now, as for the colonies, they are large. They have a narrow zone of beta hemolysis, and the preliminary identification of this species relies heavily on what we call a positive camp reaction. Next, we have your group C, Streptococci. Under this, we have your S. Disagalactiae. This organism, they produce large colonies with a large zone of beta hemolysis on your still blood agar. And infections from this species account for less than 1% of all bacteremias. Take note that members of this group, they can cause pharyngitis, even endocarditis, and as well as meningitis. However, most clinical infections from your group C streptococci, they occur in patients with underlying illnesses. Presumptive differentiation between your S. disagalactiae and your other beta hemolytic streptococci is based primarily on the resistance to bacitracin and also a negative CAMP test. Moving forward to your alpha hemolytic groups, under this we have Streptococcus pneumoniae. This organism is the causative agent of your bacterial pneumonia. They can cause meningitis as well as otitis media, which is an ear infection commonly found in children. What else? It normally colonizes the pharynx, but in certain situations, it can also spread to the lungs. Not just the lungs, pwede rin sa sinuses or even the middle ear. And virulent strains of this organism are covered with a polysaccharide capsule. Presumptive identification of your S. pneumoniae can be made with a positive optochin susceptibility test. Still under your alpha hemolytic groups, we have the viridans streptococci group. Although these organisms have few virulence factors and are constituents of the normal flora, when these organisms are introduced into tissues through either dental or surgical means, they can also cause severe infections. The most serious complication of all viridans infections is your subacute endocarditis. So what do they look like under or on your blood agar? They look very small, gray to whitish gray, and they are also opaque, having opaque colonies. The culture, no, they have rod-like and rod-like shape if you're going to take a look at them at the microscope after gram staining, and also they grow still in chains. As for testing, viridans group can be differentiated from the pneumococci and enterococci by a negative result in your bile esculin hydrolysis test, as well as salt tolerance test and the optochin susceptibility test. Now we have your group D Enterococci. There are two principal species of this enterococcal group. We have your Enterococcus faecalis and Enterococcus fascium or facium. They are predominantly inhabitants of the GIT or the gastrointestinal tract. However, they can also be isolated from the genitourinary tract and the oral cavity. The Enterococci, they do not possess many virulence factors. They are important pathogens in hospitalized patients where they can cause UTI or the urinary tract infection, even bacteremia and endocarditis. On the blood agar plate, they have very large colonies. They can also appear non-hemolytic or gamma, pwede ring alpha hemolytic or rarely your beta hemolytic. As for the culture, after staining, when you see them under the microscope, you can see diplococci in very short chains. Enterococcus faecalis, they can be non-hemolytic or beta hemolytic, depending on the strain and the type of blood agar used. But for your Enterococcus fascium, they are alpha hemolytic. Next, we have your group D na non-enterococci. Under this, we have your S. bovis. This is found usually in intestinal tracts of humans as well as in cows, pwede din sa sheep and other ruminants. Although we can find this no in many animals, the S. bovis is a human pathogen and it is known to be a causative agent of endocarditis and meningitis. It is also associated with malignancies of the GIT or the gastrointestinal tract. So what would it look like under the microscope after staining? They are usually arranged in pairs. Some are in short chains. They are large. They have large colonies, mucoid also because of the presence of a capsule, and they are either non-hemolytic or alpha hemolytic. For testing, one key reaction for this group is that they have a positive bile esculin test but negative in the salt broth test. Now, speaking of the laboratory diagnosis, no, here are the following testing wherein we can differentiate no, one from the other because these are all characteristics under your Streptococci and Enterococci. So, we will talk about it further in the following slides. So, if we're going to follow the diagram here, first, no, we're going to check their gram reaction. So, after gram staining, your Streptococci and Enterococci, they will have a positive reaction. So, they are gram positive. Again, they are cocci, similar with your Staphylococci and Micrococci. Now, since we know that all of those are similar in gram reaction, we need to differentiate one from the other so that we can eliminate these organisms for us to better identify the organism that is present on the sample. So what are we going to do next? We're going to do a test that will differentiate your Staphylococcus from your Streptococci, which is your catalase test. Why? The one organism that will have the enzyme catalase is your Staphylococci. So, kung walang enzyme na catalase yung Streptococci, the result of that test would be negative. So, catalase negative, we know that that is already a Streptococcus or Streptococci species. So, now, we need to know the specific organism under the Streptococci, no, na family. So, what are we going to do next? First, you need to take a look at the colony. You need to take a look at the culture media. Check the hemolytic pattern if they are under the alpha, beta, or gamma hemolysis. If they are having alpha hemolysis, no, the next thing to do is to what? So here, alpha hemolysis, you're going to proceed with Optochin testing. Optochin, this is an antibiotic, so the positive and negative reaction here is known to be susceptible or resistant. Susceptible or sensitive or negative resistant no to the antibiotic. If that organism that you have isolated is sensitive to the optochin disc, then therefore we can conclude that that is the Streptococcus pneumoniae. But if that is resistant, we need to look for further because maybe it will be under a very dense Enterococcus or other group D Streptococci. So what are we going to do next? Bile esculin. So for bile esculin, no, the positive reaction and negative reaction here, if it is negative to bile esculin, then that is a very dense Streptococci. Pero pag nag-positive, do further testing. Kailangan muna gawin natin yung PYR testing or testing that will involve application of the 6.5% sodium chloride. If that is positive for the test, we can conclude that it is one under the Enterococcus species. If that is negative, that is one under the group D, Streptococci. Now, for the specific procedure, first we have your optochin or the P-disc susceptibility test. The purpose of this test is to determine the effect of your optochin also known as your ethyl hydrocuprane hydrochloride, on your organism. Optochin mainly lyses the pneumococci. Pag na-lyse, that is a positive reaction. But alpha streptococci are resistant. So, hindi mal-lyse no, yung ating alpha streptococci, leading to a negative test. Now, for the method, first, you're going to get your inoculating loop. Mag-streak tayo 2 or 3 suspect colonies of your pure culture onto half of your 5% sheep blood agar. So, the media that you're going to use here is your 5% sheep blood agar. Then, using your heated forceps, after sterilization, you're going to get your disc, place it on the upper third of the streak area. Gently tap the disc to ensure adequate contact so that after inversion, no, hindi malalaglag yung pinaka-disc mo. Afterwards, you incubate it first. Wait for 18 to 24 hours. It should be at 35 degrees Celsius and dapat po with 5% carbon dioxide. Take note that cultures do not grow as well in ambient air and they have larger zones of inhibition. Afterwards, after po natin mag-incubate and you see a zone of inhibition like this one, no, you have to measure in millimeters, including the diameter of your disc. Now, what will you expect no for your results? Pag positive yan, ibig sabihin, yung zone of inhibition mo, nag-measure ng at least 14 millimeters. At least, no? So, this is the minimum. So, any within 14 greater than or equal to 14 millimeters is a positive or sensitive or susceptible to your optochin. Negative would be meaning walang zone of inhibition. Now, take note that pneumococci, they are sensitive and viridans natin, they are resistant sa ating optochin disc. Next, we have the bile solubility test. This test mainly differentiates your Streptococcus pneumoniae, which will be positive, from your alpha hemolytic streptococci na negative, meaning insoluble siya. Principle, your bile or a solution of bile salt, example, sodium deoxycholate, they rapidly lyses pneumococcal colonies. The lysis will depend on the presence of an intracellular autolytic enzyme, which is amidase. Bile salts, they will lower the surface tension between the bacterial cell membrane and the medium, thus accelerating the organism's natural autolytic process. So, how are we going to do this procedure? First, after 12 to 24 hours of incubation ng ating media, which is 5% sheep's blood agar, we will place 1 to 2 drops of 10% sodium deoxycholate on a well-isolated colony. So, example, meron tayong colony like this, no? So, the colony, papatakan po natin siya ng 1 to 2 drops daw ng 10% sodium deoxycholate. Take note, a tube test is usually performed with 2% sodium deoxycholate. Now, afterwards, you gently wash the liquid over the colony without dislodging the colony from the surface of the agar. Then, we incubate the plate at 35 degrees Celsius up to 37 in ambient air for just 30 minutes. Take note kung malalays ba or masisira yung colony. Positive, of course, madidisintegrate, masisira yung ating colony, the colony will lyse, then an imprint of the lyse colony may remain in the zone. Negative, kailangan buo pa rin yung colonies, bilog din siya. Kaya, as you notice here, yung letter A natin, nawala na, na-lyse, no? Imprint na lang siya dyan. Na-lyse na po yung colony, wala na yung pinakabilog as compared to letter B, nabuo pa yung circle, no? Buo pa, intact pa yung colony. Next, we have your bile esculin testing. What usually happens here, yung organism mo kasi dito, no, you want to differentiate, you want to identify if that is an Enterococcus or if that will be under the Streptococcus bovis group. So this test will mainly differentiate your Enterococci and group D Streptococci from the non-group D na viridan Streptococci. So usually you have your organism, yung organism mo, it will grow in 4% bile that will be able to hydrolyze the esculin to esculitin. Esculitin naman will bind, no? Will bind with your, or react rather, with your ferric ions. And then that reaction will give you a dark brown to black precipitate. Now, take note for your BE or your bile esculin hydrolysis testing, all group D streptococci, they are positive for this testing. This is known to be the best physiological test that we have for the identification of your group D streptococci. Both enterococcal and non-enterococcal species of your group D, they are able to hydrolyze esculin in your agar slant, causing the slant to darken. But take note that if less than half of the slant here is blackened, if less than half of the slant is blackened, or if no blackening occurs within 24 to 48 hours, then, katulad nitong nasa letter B, the test will be reported as negative. So, ano ba yung mga slant na yan? Bakit may blackening? Paano mo po yan muna magagawa? Take note of the method. You need to inoculate 1 to 2 colonies from your 18 to 24-hour culture. Lagay po natin, streak po natin sa ating slant of the tube media. Afterwards, incubate lang natin at 35 to 37 degrees Celsius. Take note of ambient air for 48 hours. Again, pag positive, may growth, may blackening of the agar slant. Negative naman if less than half nung slant and no blackening of the medium. Next, we have your salt tolerance test. From the name itself, you're going to use here the mechanism where the organisms will be checked if they will grow in high salt concentration. Now, we do this to differentiate your enterococci, which will be positive for the test, and your non-enterococci na magiging negative. Media to be used here is your heart infusion broth containing 6.5% sodium chloride. We also have here an indicator, which is your glucose and bromcresol purple. They are known to be indicators here for acid production. Now, since salt tolerance, titignan mo yung organism na tolerant sa salt, sa high concentrations of salt. So, sino ba ang mabubuhay? Tolerant ba siya? That would be your group D enterococci, and ang hindi naman tolerant would be your other group D. No? Hindi sila tolerant, so hindi sila mabubuhay in high salt concentrations. Now, how do we do this procedure? First, mag-inoculate muna tayo. You get 1 or 2 colonies from your culture, 18 to 24-hour culture. Lagay po natin diretso sa 6.5% of your sodium chloride broth. After we have inoculated that, we now incubate. Still 35 to 37 degrees Celsius, present in ambient air for a total of 48 hours. Now, all enterococci of group D, they will produce heavy growth sa 6.5% sodium chloride broth. But your S. bovis, they will not grow in this culture media. So, this test will provide us with a good method for differentiating your organisms of the group D. Again, pag positive yung result, ibig sabihin may mabubuhay. So, indication that there is bacterial growth in our sample ay merong turbidity. Kailangan may turbidity within 48 hours. And you will notice that there would be a color change from purple magiging yellow. So, that is for a positive reaction. But if the tube is negative at 24 hours, incubate nyo pa din ulit. After incubation, you check it again after 48 and 72 hours. If the organism is salt tolerant and BE positive, it will be considered to be an Enterococcus. Now, here we have a diagram for the differentiation of your group D Streptococci from Enterococcus. Again, both they may show alpha, beta, or no hemolysis on your blood agar. But what you can do to differentiate, check mo pa din muna yung bile esculin. If nag-positive po pareho, then it means it's one or the other. So, how are you going to break the tie? Kailangan po gumawa tayo ng 6.5% sodium chloride testing. Ang mabubuhay will be your Enterococcus PC. Ang hindi ay Group D Streptococci. Next, we have the schematic diagram for the differentiation of your gas or the Group A Streptococci from your Group B Streptococci. So, first, let's just start with your gram-positive reaction. Gram-positive daw siya na cocci, tapos catalase negative pa. We know that this is a streptococci, no? So, what are we going to do next? Let's check the bacitracin test. Bacitracin susceptible, they are under group A. Pag resistant, you do CAM testing. Ang magpa-positive dyan would be your group B, which is Streptococcus agalactiae. Negative naman, it's positive, possibly a group D or a beta hemolytic streptococci. It's not under your group A, B, or D. Now, how do we do a bacitracin testing? So, I know you have already done this during staphylococci identification, but this time around magkakaroon lang ng iba because it will have different oxygen conditions or air conditions na gagamitin sa testing. So similarly, we're going to use your inoculating loop, mag-streak tayo 2 or 3 suspect colonies of a pure culture directly on your blood agar plate. So, ang median natin dito is still your 5% sheep blood agar. Antibiotic of choice is still your Taxo A or the 0.04 units of bacitracin. So, pagkalagay nyo po ng antibiotic this dyan, of course, you need to incubate. Upon incubation sa 35 to 37 degrees Celsius, kailangan po ilagay natin sa 5 to 10% carbon dioxide concentration if that will be for streptococci differentiation, unlike sa staphylococci na ambient air lang. And then afterwards, you look for a zone of inhibition. Now, positive or sensitive siya if the zone of inhibition is greater than 10 millimeters. For your CAM testing, this is also known as your Christie, Atkins, and Munch-Peterson test, which is used to differentiate your Group B Streptococci. Group B is your Streptococcus agalactiae from other streptococcal species. Also, Listeria monocytogenes also produces a positive CAM reaction. Culture media natin dito will still be your 5% sheep blood agar. What happens here, meron kang CAM factor. This is a diffusible extracellular hemolytic protein. It will combine with your beta-lysine of the Staphylococcus aureus, and the result will be an enhanced lysis of red blood cells. Yung enhanced lysis na yon, no, it appears as an arrowhead zone of hemolysis adjacent to the place where the two streak lines come into proximity. So, anong ibig sabihin nun? Dito kasi, no, sa methodology natin of your CAM testing, first, ang gagamitin mo, magsistreak ka ng beta-lysine producing strain of your S. aureus. So, ibig sabihin, capable siya dapat mag-lyse because you're going to use that as an indicator. Papunta doon yung organism mo. So, magsistreak daw muna tayo, no, ng line nasa gitna, one streak of your S. aureus na nagkukos ng beta hemolysis. Afterwards, you're going to streak your test organisms across the plate. So, yung streaks natin dito, yung dalawang streaks, ito yung organism mo. It should be perpendicular, no? Perpendicular to the streak that you use for your S. aureus. It should be within 2 millimeters. So, hindi siya touching, but it's close, no? Almost close to touching. Take note that multiple organisms can be tested on your single plate. And then afterwards, incubate natin overnight at 35 to 37 degrees Celsius. Anong magiging positive reaction? So, ang positive reaction natin dito is your letter A. So, nagkakaroon siya ng arrowhead production, arrowhead-shaped zone of your beta hemolysis na katuro papunta doon sa ating S. aureus streak. So, it will be formed at the juncture of the two organisms. Next, we have your hippurate hydrolysis, which is also useful, no, in the presumptive identifications of many organisms. Now, the enzyme responsible here is hippuricase. Now, in the presence of hippuricase, hippuric acid will be hydrolyzed, forming your glycine and benzoic acid. Glycine is deaminated further by the oxidizing agent, which is ninhydrin. This is reduced during the process, and your end products of the ninhydrin oxidations, they react to form a purple colored product. Take note that this test medium must contain only hippurate because ninhydrin reagent might react with any free amino acids present in the growth media or other broths. How do we do this testing? First, we're going to add 0.1 ml of sterile water to a 12 by 75 millimeters plastic na test tube. Afterwards, you make a heavy suspension of your organism to be tested, and using your heated forceps, you place a rapid hippurate disc in the mixture. You cover or cap, no, incubate natin yung tube for 2 hours at 35 degrees Celsius. Use of a water bath is preferred for incubation rather than using the incubator. Now, afterwards, you further add 0.2 ml of your ninhydrin reagent and then re-incubate pa natin 2 or 4 an additional 15 to 30 minutes. Then, observe if there would be a color change. There would be a development of a deeper color po. That would be a positive result. So, positive result, deeper color. Negative naman po natin, it's either yellowish, slight yellow, pink color, or colorless. So, reagent natin again is just your 0.2 ml of ninhydrin dito. So, just a brief summary, naka-table form, no, yung diagram natin of your identification ng gram-positive cocci. So, you start first, no, with your gram stain reaction. Afterwards, since gram-positive cocci, may iisipin nyo dyan if that is Staphylococci or Streptococci. So, first, differentiate lang muna natin. So, ang main objective muna natin dito is to identify the Streptococci organism. So, let's assume that the catalase reaction is negative. So, Streptococci na yung genus. Let's now check no kung ano kaya yung ating um, species. So, first thing to do is you check. Check natin yung hemolytic pattern. Beta ba yan, alpha, or non-hemolytic. And then, you follow the diagram down until you reach your specific organism. Now, as for the last slide, no, it just shows here its different categories and also the following reactions to certain testings to further help you in checking if that would be under and further identify no, your specific organism.